Hi, everybody. Let me get moving and grooving. All right. So anyway, getting back to once we've opened Eugene, let's get that one shut. It's take a little while. So if you downloaded Eugene and extracted all the files, you should find right at the bottom the Eugene UI. That's the one you want to click on. Sure. And use. So you can use that just from the folder that you extracted all of that stuff to. So let's get chat back up. So again, kind of download Eugene, extract the zip to a folder, click on Eugene UI at the bottom. And that's Eugene. Okay, give people a moment to do so. Back to is it starting up okay for you, Adam? Cool. Yeah, it's kind of part of the, this isn't necessarily a particularly user-friendly piece of software. Actually, you know what would be useful? Give me just a second. You can get a rare look at the inside of Blackboard from my perspective. Just double checking something. Okay, that's a good one. Just wanted to double check that before I uploaded it. There you go. That's something else you can use if you wish. So this is the download of the wild type version of the yellow gene sequence. Robert, that kind of depends on, is that talking about downloading to computer or extracting the files? Oh yeah, that'll take a while. I went and had my lunch while mine was doing it. So it may take a little while longer, but probably shouldn't be more than, another 10 minutes or so. But it'll be around for us to use hopefully today.
So
But
I
again,
tried downloading the wild type file that you had right there,
uh-huh.
but it wasn't letting me be able to view it.
So when you open Eugene, save it to your computer, same folder that you're doing all the other sequence analysis stuff in, and then try opening it from there using Eugene. Yeah, it may take a little bit longer, Robert. Again, don't worry about it too much. We have the freedom to do more of this on next Tuesday. So, keep watching. It's not going to hurt you when it comes to doing this for yourself, right? And then we can always go through it again on Tuesday as necessary if people need more time and more support. Did that do the trick, Adam?
No, so I tried saving it to the folder that I have,
Hmm?
but then it just says, oh, give me an option of how I would like to open it. And it says, how do you want to open this file? Adobe CC, Word, Notepad.
Okay, so that's because you haven't used Eugene on your computer yet. So
Mhm.
which one was this? I think this was the new one, wasn't it? Maybe. Anyway, get rid of this. Only helps useful to have one open. So I'm just going to delete that off your trot. Okay, so go to Eugene, Control O, or you can go up to File and Open. Then navigate to your folder that you have everything stored in. In which case mine happens to be the lab, right? Because that's where everything's at. And then find that file. In my case, I've got a whole load of them because I tried on doing different things with it. And click on it, just go OK. And that should open up that sequence. You just kind of play around with it as you see fit. There's all kinds of funkiness. And I got to tell you, I don't know half the things that this thing can do. Not even close. That wasn't very useful. Anyway, did that help?
I'm still trying to figure it out because it won't let me save into a folder or anything.
So when you go to Blackboard, you can't save it to a folder?
Correct. I can only show it in a folder, but not save it. And even then in the show, it doesn't pop up.
Hmm. What's this look like in student view? So hang on a second, Dipper. I'll just get to. So click on it. It'll throw a fit because it doesn't know what to do. You just click save. This is in the same folder. That should save it to that folder, Adam. And then you go back to Eugene, click open, and there it is. You just gotta click on that and you should open it. What part of that isn't working for you?
Just the whole save portion, because every single time I click on the hyperlink, it doesn't prompt me to the option of saving it.
What about this? If you click on the blue lettered bit down here.
Yes,
So if you click on that, it doesn't give the option to save it.
correct.
Oh shoot, I don't really know then, to be honest. Don't worry about that bit. Can you at least download the yellow sequences, that folder, the zip file?
Let me try.
Because you'd be kind of screwed if you can't. Okay, while you're doing that, Dipper, right, so have you extracted everything to your computer? Or even have you been able to open Eugene itself? Okay, cool. So that's great, Dipper. That's all you need to worry about for now. We'll actually take you through. Oh yeah, yeah. Don't worry. I'm pretty similar. I don't know what half of that stuff is. So don't worry about that. We're going to take you through each bit, kind of very carefully, step by step. Adam, that's great. Get that unzipped into that folder. In fact, all of you, if you could download this yellow sequences folder and extract that into whatever folder that you're going to be using for this project. And let me know if you need any more time. Yeah, Dipper, it's a big, scary looking program, and I'm sure that as and when I need to do, work of my own on it, I'll need to spend some time figuring out how to use it. Cool, cool. Okay, so let's get out of preview. Beep-a-doo. Yeah, don't worry about the other file. That was be nice if you can have it. You should already have a genomic file already in that folder from Tuesday. So the file that you downloaded from the NCBI website is essentially the same, but it's the yellow 1 mutant allele. So if you just keep that in mind, when we get to it, then not really any difference, to be honest. Does that make sense? Cool. Yeah, the only difference is the A to C transition. Okay, so as I did last time, I'm going to walk you through this using the handout so I don't take any shortcuts that are unhelpful. Pardon me. So you should have done already all of this stuff, right? And the hope is that you've also done the questions leading up to this point in the assignment too. Don't worry if you haven't, that's fine as well. I mean, all this stuff is, you know, online anyway, but you know, it's good to kind of get stuff done as you go. Hey, look at that, it takes me back to where I was. What was question 9? Ah, there we go. Okay, so this is basically the first question. Oh, I'll be sleeping on my leg again. The first question of like the Eugene really specific stuff, right? And so we're going to pick up there. And what we're going to do first is we're going to actually pick one of those sequences from the good yellow sequences folder. Sorry, this puppy loves sleeping upside down. It's very distracting. Okay. I'll try and avoid showing my feet, so. Where is she? There she's at. She's a funny dog. Dog with 9 lives, of which she's already used about 4 or 5 of them. So hopefully she's going to lead a pretty sheltered life from now on. Anyway, so what we need to do now is use Eugene to open one of those files. So again, you can do that through the file drop down and click open, or you just do Control O. Right, so where did I save all of these things? I've got a really messy folder here. Here we go. Okay, so all of these are sequence data files, ABI files. So you can pick whichever ones you want. Doesn't really matter. You don't have to work with all of these either. They're just a selection of files from both mutant flies, so yellow-bodied flies, and wild-type flies. I don't know which is which. So let's just pick one of them. All right, so where's the actual sequence file? So this is one of the times that I go like, ah, shit, Eugene, you're such a pain in the ass. And nope. Nope. There it is. Right. So you might have to do a little bit of fiddle farting around to get it to show what you want. That's fine. Along with, you now know how I approach things that I don't understand. I just hit like buttons in turn, and eventually it works, right? It's not actually how I drive cars, thankfully, or, you know, operate machinery, but it's worked in this case. Okay, so open up that file, and the hope is for as many of you as possible to do this as we go through it, right? Because it will make it a little easier, give you a little bit of a time opportunity to go, Dr. Crook, this program's a piece of shit. And I'll be able to say, no, it's not. And hopefully take you through how to kind of work with it a little bit better. So this is like super zoomed out. Let's zoom in a little bit. Do that. There we go. I was about to swear and I didn't. I'm very proud of myself. So this is do hickory do, which will allow you to scroll through the sequence. And it will show you what bit of the sequence you're looking at up here. Right, so we're looking at about the first 40-ish base pairs, which is true, of the sequence. And you'll see that this looks like complete ass, right? It looks terrible. I shouldn't say stuff like that. Kind of slip into the vernacular, unfortunately, sometimes. Notice I said ass, not arse. So I'm becoming more American, which is maybe a little disturbing. No, it's a bad thing, but anyway. So what you'll see is that the chromatogram, the electropherogram looks like crap, right, at the beginning. And it's only when you get to about, I don't know, about 25, 30 base pairs that you start getting a nice, clean signal. And you can see it actually improves for a bit, and then it'll start getting worse. Probably not, yeah, this looks a bit crappy, but this isn't a long enough bit of sequence to start getting into issues at the other end. That's typically around 700-800 base pairs when quality degrades. But at the beginning, what happens is essentially it's very hard to resolve like itty bitty teeny tiny pieces of DNA very well. And so the very first ones that come off the capillary sequencer off down that tube are the smallest because they're the ones moving the fastest. And so it's really hard to resolve those. So for the first, few until you get like reasonable sized chunks, you get really poor quality sequence. So all of this is not really any use, right? So we just kind of discard all of this. So the first thing to do is, so right click on the sequence view pane, select edit new sequence. So right click, edit new sequence. Oh yeah, Valeria, Eugene is a bit of a beast. And then click create. Oh, let me get back to where I was at. Give me just a second. Oh, here we go. This is the new one. Okay, let me think. Okay, Adam, you're going to need to take me through a little bit of what you're doing and so I can help you figure out what's going wrong. So what are you talking about when you say downloading the report? You can unmute your mic as well. It might be easier than doing it all by chat.
So that's where we had to go like to the top right-hand corner and click on send to, and then create file, and then it gave us the automatic option to have it as a report.
Hang on. So this is back in the NCBI.
Yeah. And I had created a folder and named it, and then I downloaded the file from NCHBI or whatever. And then it just pops up as a text file.
All right, give me a second. So that's here. So you went send to?
Yes.
And clicked file?
Yes.
You create file?
Yes.
So I got to put me back over somewhere else. You get back over there. Right, so I've saved bleh, GB, GenBank file. Now,
but like for you, you know how whenever you clicked on send to and then you made it as a file, and then it gives you an option between GenBank or something else.
Yep.
It didn't do that for me as a format. It just says full report as a text, gene table as a text, summary text, tabular text, ASN1, XML, and UI list.
That's weird.
Yeah.
you're not like using Netscape Explorer from the 1990s, are you, by any chance?
No, I'm using Google Chrome.
I don't know why it's not allowing you to do that. Are you doing it through the like all the steps that I just showed?
hmm I can share my screen if you want me to.
I'm not entirely sure how to do that, to be honest. I'm not much of a computer whiz, to be honest.
Oh, okay.
So, let me think, let me think, let me think. So, do you have selected region selected here? Just make sure you're not like downloading something different.
No, I do not.
So make sure it says selected region. Because you don't want to download the entire X chromosome. That would not be a bunch of fun.
Was it the Y yellow, the name of it that we were choosing, or the Drosophila melanogaster chromosome X?
So you need to
because I used gene databases, and then I typed in what you had in the Word document, and I clicked on the Y.
mhm.
And then from there, I was able to see the genomic regions, transcripts and products and all that stuff. And then I went to file.
So have you gone to NCBI reference sequences? This bit down at the bottom.
Yeah.
Okay. And you clicked on down to the reference assembly where it says genomic. You've clicked on the GenBank link.
Okay, now I did it. I clicked on it. And then send to file, format GenBank.
Create file.
But now it says, how do you want to open this file? And I can't save it.
Does it not bring up this?
No, it
Like a
just
save as
has a
file?
dialog box saying, how do you want to open this file? Look for an app in the Microsoft Store. And it's labeled as a GB file.
Yeah, I'm not savvy enough to know why your computer's doing that.
Oh, okay.
Normally there's like a default kind of download folder for Chrome.
Okay.
Okay. Grab it, love. She's gonna jump. There you go. Yeah, Dipper, if you disappear, that's totally fine. We can always catch up another day too. If that happens. Hopefully it won't, but if it does, it does. Yeah, I don't know, Adam. I'm sorry. I can't really
So it's all survival.
I don't know why you wouldn't be able to save something from a hyperlink in Chrome.
Okay. Because everything's all up to date.
I'm more really, love. You got to get her off. Got to get her off, love. Sorry, we can't let our puppy jump off the couch because she's might tear open her stitches.
Oh, okay. Well, I guess I'll just go ahead and have to make my best educated guess for this.
Give me a second.
Okay.
Actually, do you have like Microsoft Edge or anything like that, another browser installed on your computer?
Yes.
Try that. That's usually my answer to most things. And then see if it works the way it should, using the exact same link and information. Pardon me, same process. And fingers crossed it will work with that, because sometimes Chrome might be doing something funky. Valeria, have you extracted that file, the GenBank download, already?
I think so. So I was able to download the other two, but it says that Eugene just isn't responding every time.
Oh, okay.
So I'm gonna have to delete it and re-download it.
So once you've
I just
downloaded the file, you know, when you go to your downloads,
Mhm.
let's go back a wee bit.
I just tried it on Microsoft Edge and it's still the same thing.
Oh, hang on, just give me a moment, Adam. And so what you should have a folder that looks like this, that when you click on it, it says extract all up at the top. If you have a PC, if you've got a Mac, I don't really know, because I don't have a Mac. And then when you click extract all, it would take you a while. Like, I don't know if Roberts has finished yet or not, hopefully. But it'll take a while for it to extract all of those files. And when it does, it will look something like this, right? You'll click on it, and way down at the bottom, that one there, Eugene UI.
Mhm.
Yeah, that's the one. Click on that, and then that should, if it has enough time, open.
Okay, let me try that out real quick.
It may be that you won't be able to get it all extracted and opened and everything in time for using it today.
Yeah.
But then we'll just go through the same stuff, if necessary, on Tuesday.
Okay, sounds good.
Yeah, so if it's not working for you today, don't worry about it. Just make sure you have it for Tuesday.
Okay.
And we'll kind of use that lab session either for anything that we didn't get done today, but also for everybody, for anybody that isn't able to do it today because they couldn't get Eugene open and ready.
Okay.
Cool. Yeah, Adam, I mean, if you can't even download a file from Blackboard, I'm kind of a little bit stumped, to be honest. Because otherwise, gee, I wonder if I can put stuff up on Dropbox. That might be a way of doing it. Where we be at? We be at here. Yeah, I don't know. I don't know why. Class activity to work on the lab together. I don't understand, Dipper. How do you mean? Where's Dropbox? Give me just a sec. Give that a shot, Adam. See if you can get it from there. Well, I mean, I'm going to go through the lab from the handout, essentially. so we can all go through the different exercises. And then if you're not able to do it with me as we go through, then we can just do that on Tuesday too. So you'll all be able to get it done. I'm not particularly worried about that. Plus this assignment isn't due until next Thursday, so there's plenty of time to be able to figure stuff out, basically. Does that help?
Oh, no, it's not.
Oh,
It just says .fa files can't be previewed.
that's fine. All you have to do is,
But I clicked download, direct download, and it didn't work.
and it
It's
didn't.
the same thing.
There's something funky going on with your computer, I'm afraid, Adam. I don't really know what. Do you have like downloads from suspicious looking English people blocked or something?
No, I don't. I only use my computer for school purposes only.
So do we just need this program to really just help us answer these questions, or are we doing something else with it with this program?
No, we're actually going to work through the analysis. You do need it to help answer the questions because the questions are tied to you doing specific things with the program. And so the problem we're having with Adam is for some reason he can't get the wild type or the genomic sequence for some reason. His computer's not letting him download it from any source. Shark, I wonder if I could even just send it to you as an email. What's your email address, Adam? We'll get there eventually, don't worry.
RMON.
Oh, there we go.
Yeah.
Something's got to bloody work, right? Otherwise, I'll put it in a letter and I'll post it to you. Pepper, be quiet. Okay. You can probably hear my dog in the background. All right, give that a shot. Something's got to bloody work. Oh, now we've got the printer going off too. Okay, what was the other
Yeah,
comment?
I agree with that. And I thought I was thinking out loud or something. I was like, oh shoot. So, I was hearing the dog.
Yeah, it's not your subconscious screaming at you to choose a different career path or something. No, we're having lots of other issues with the lab and programs and files and stuff. So one of my students thought that was just his brain screaming at him. It's like, no, it's the dog. All right. So anyone else having issues with Eugene or getting the genomic sequence, or even getting the? Can't you just save it from your, like, right click on it from your email program and save?
I tried.
Does the right click button work on your computer? Maybe that's it.
Yes, it does. There's preview, open, quick print, save as, hit
Save as.
and then it says the file name, but save as type FA
Oh,
file.
hang on, I didn't hear that. My dog was whining. Say again.
It has the file name listed and then the save as type followed as FA file, and I hit save.
Right.
And then I go to that file and it pops up as a text document. And then
Now,
FA
have you
file.
opened that from within Eugene? Right,
Let
so
me
you have the file saved to your computer, right?
Yes.
Okay, so that file is in a format that reads as a text file if you just open it with a web browser, for example, or if you open it with Windows Explorer. You have to open it with Eugene for it to work properly in Eugene. So open up click on your Eugene app.
Okay, it says the document selected contains multiple sequence instances. Please select the way Eugene will read these as a separate sequence in Sequence Viewer.
Yeah, just go with the default. Click yes.
Okay, so now I'm looking at all the nucleotides, but I don't know if it's the right one.
Yeah, that's what it should look like this, right?
Uh,
Just a whole sequence of letters.
yeah,
Yay! Woohoo! Happy
no,
dance! Happy dance! Okay, great. That's all you need to do for that. You're good. So now what we need to do is, sorry, I should put this down as like my greatest victory in helping a student in an online class. Talk about it in future job applications or something. Anyway, so now what we need to do is we need to open a sequence file. I don't know why the electropherogram has disappeared. Let's try this one. Nope. Come on. There we go.
okay, but how did you get to the A, F, C, Q, or G?
Okay, so same as always. File, open, and those will be in your good yellow sequences folder. So this is what I mean by saying that, and this not picking on you in particular, Adam, at all. Clearly something like funky is going on inside your PC for some reason. I don't know what, but this whole kind of lab in general, it works really nicely, but you have to follow the instructions like to the letter almost. If you do, and it might feel a little bit like, what am I, some kind of simpleton kind of deal? But if you do that, then it will work like a dream. Things go off the rails a little bit if you try kind of doing things a little differently. Not saying that that's what's been happening here, but just saying in general, this is what you need to do for this to work. So you have to have those yellow sequence files extracted. You can't work on them when they're still in the zip file. You have to extract them and open one of them. And you can open whichever one you wish. It's complete like lucky dip essentially. Doesn't matter. I just picked the first one because I don't have much imagination when it comes to picking things. It's looking a little bit gray out there. What if we're going to have any rain? I'll have to get the kids to get their washing in. All right, so once you've opened one of those, you should have something that looks like this, maybe with a bit of fiddling. And when you do, hey, right click on it. Oops. Let's try again. Right click on it and hit edit new sequence and just say create. That'll open up this sequence, which you can modify without changing the one which was open before. All right. So now what you can do is you have these two sequences up here, right? The original sequence and the top one, which is the one that you're going to edit. If you look at it originally, oh, it's actually in edit mode, so it will do it for both of them. Now you can actually click on one of those letters and you can change that sequence to something different by looking at this sequence. So if you looked at this, it says that it's thymine, but you look at that, it's like, don't think so. Is it raining out there? What about all the washing? Thanks, love. So these are clearly not good calls, right? They're not accurate sequence. Oh, the washing. Hey, love, can you help Evie get the washing in, love? Oh, it's in? Oh, thanks. Phew.
How did
Started to
you
rain.
change the display of the top where all the lines are, the wavy lines? Or
To
sequence, I
make it
guess.
bigger?
Yeah.
So you see these tiny little icons down here?
Mm-hmm.
You want to use that. So if you zoom out, it'll look, typically it opens it looking like this.
Mm-hmm.
Click on the plus icon until you're happy with it. I think that's,
Okay.
crikey, can it go even further? Well, that's no bloody use, is it? There you go, that's better. Can't believe how much it's rained this summer. Yeah, exactly. Bloody mosquitoes. I think Mama said she got it in already. Oh, great. Thank you, love. And so kind of go back to where we're at the beginning. Now, once you've hit edit new sequence, wow, since 1972, that's officially before I was born. So that's a long time ago. I am old. I am ancient. And I should be venerated as such. One of your living ancestors. Okay, so if you click on one of these, you're like, well, that's clearly not a T. That's just, that's not really sure what that is actually. So you go through and you just do that for all of the ones which look, suspicious. I'd probably do it at least to the start of those T's.
And then how did you get the nucleotides on the top other
Okay.
than the ones that are already there?
So you right click on the pane itself and then select edit new sequence from the drop down menu. You see that?
Oh, okay.
Right, so go through and do that. And then you should be able to save this to your folder. Now, it says to do that for every sequence in the Eugene folder. I think that would take you a very, very long time. So I'd suggest doing this for 5 or 6, should give you a decent sample size. All right, so you can save that simply by you can save, new project. So if anything happens, like the program crashes or whatever, you'll be able to open it back up where you're at. So now if you want to work on a second one, open again. And so you can see this is the new edited sequence that I've been making, right? That's just been automatically saved. Let's go down here and do whoever TF and DR were. You do the same again, kind of zoom in a little bit. This is another pretty nice looking sequence. You can see the same thing again here, where at the beginning of the sequence, it just looks like junk. And again, right click, edit new sequence, just click create, and then go through and change all those dodgies. Oh, that's already an N, don't need to change that. Change all of those to N's. N basically means any, right? So you're not making a judgment as to what base that is. It just could be any base. We don't know. Right. And then that should save that automatically. Yeah, it saves that automatically. Okay, does that make sense, everybody? Now you can answer question 9. Where has that gone? So this is what I mean, Robert, by you can technically try and do this assignment without doing any of this stuff in Eugene, but it's not going to be necessarily easy to understand what is being asked of you. Right, so this is a pretty straightforward question. It even tells you what it is in the handout. So in this case, you know, not too big of a deal, right? For the other questions, it will make a difference. Okay, so the next thing, so there's 2 more things we need to do. One, we need to figure out where in our sequence that, in our reference sequence, right, the original genomic sequence, where does our little bit map to, right? Now we think it should be where we design the primers to, because if PCR worked, then presumably, you know, we did a good job. But we can actually do that formally by using Eugene to map our sequence to our reference, right, which is our genomic sequence. Okay, so what we need to do is make sure we have a reference sequence. Now this could be the one you downloaded from NCBI. It could be the one that you've downloaded from me uploading it to Blackboard, or, you know, whatever means that we've used. So you need to have that open and have done a few of these sequences, right, changing them. So now we want to map our our sequence. So you go on to yeah, that's weird. Tool setting, Sanger data analysis, and then map reads to reference. Right, this will bring up a kind of an instruction box, like where you tell it where to look. And so you need, yes, that's correct, Adam, 10 points for both lab and for lecture. So 20 points in total for the course, essentially. Because they're separate SRIs, you're doing two lots of SRIs, get 2 lots of extra credit. So you need to tell it where your reference sequence is. So this goes back to your folder that you're working with. And I'm going to use this one. You can also use the one that you downloaded from NCBI too. Doesn't really matter so much. Now, what you're going to do is you're going to upload kind of to this program the reads that you want to map, right? So now if you click on add, and I need to go back to my good yellow sequences file. Doo-doo. Now you want to add the ones that you've cleaned up. Oh, there's only one of them. That'll do. Don't change anything else. Just hit map. And it will... Oh, fudge monkeys. Okay. I remember what causes this. Give me just a second. Doo-doo. Okay, so you just need to rename this to remove the underscores, the underlines. For some reason, which kind of doesn't make any sense to me, given that this is a bioinformatics software, it completely flips its wig at underscores. Okay, let's try that again. So tools, Sanger data analysis. Let's see, it should do, but because I remember I have this problem every single semester, believe it or not. Actually, I really need to change that handout to reflect that. That's a good point. Let me make a note of that. Otherwise, I've got memory like a fish. I'll just forget it. Ah, I didn't do it again. Fucking hell. Pardon my French. All right, so give me just a moment and I'll dig it out again. Only take me a minute or two. Oh, okay. It's also the semicolons. There we go. Shit. I'll write this down. There we go. Remove. I shouldn't have any of that in there. The rated chromatogram is not found. Oh, come on. Yay, 4th time's charm. So instead of using any of the files that you have already played around with, it's going to make things a little noisier. Oh, hey, let me go back to sharing. There we go. So I'm just going to do this again so you can see what I did. Okay, so saying a data analysis, map reads to reference. Pick your file and then add one of the original file that you're working on. Right, so I was working on this AFCG file. So click on the original one and then click map, and ta-da, it goes all the way through. For some reason, when you edit a sequence, it strips out the chromatogram, which Eugene has a conniption over. So give that a shot, Dipper, and that should fix the issue. Robert, were you able to extract the Eugene files and open it up?
Yeah, I did, but I have an old computer, so sometimes if I have too many things open up at once, I'm afraid to have too many things open up because it'll kind of like crash and turn off and have to restart. So after this, I'm gonna have to rewatch some of this and kind of play around.
I'm not sure you'd want to rewatch it.
I mean, I didn't say I wanted to or not. But you do what you got to do, right?
Okay.
So, yeah.
So Dipper, can you see down at the bottom, there's a little kind of process box. And you can see the thing that is causing problems with mine, more than one sequence in reference file. Okay, so take me through exactly what you're doing. And you can unmute as well. It may or may not make life easier. So there's two issues. One which we shouldn't have because these file names have already been edited. Oh, okay. Yeah, no worries. So one issue is having a semicolon in the file name. That causes it to wig out. The second appears to be on those edited files. They don't have the chromatogram, which is the problem that I have if you kind of zoom in down here. So don't know why that would be the case, but so just double check, make sure there's no semicolons in any of those. Doesn't look like there is. How are you doing, Adam, and everybody else?
Very funny. I'm still trying to take everything in and stuff, and I have so many things open, and right now I just feel like so behind and a lost cause for this. And
Yeah, don't worry about it. I mean, I was in your position when I learned how to do it. It just takes a little bit of time.
Yeah.
It's true. I mean, I didn't use this before teaching this course. The key is always just to do exactly what it says in the handout, and mostly it should work, unless like weird stuff pops up.
It
So
was at the beginning, but now it's not.
so Dipper, you should get something that looks a bit like this, where you have a series of letters at the top. And if you scroll to the right using this bit, Valeria, there should be a oh, hang on a second, Dipper. Valeria, there should be a version for Eugene, which is a little more problematic, but should work too. Generally, it seems to be a bit more reliable on PCs, to be honest. I don't know why. Okay, so Dipper, hang on. You've got, so I think you're adding the wrong sequence to the wrong place. So what you need to do is you've got to add two different sequences. One is your genomic sequence. Give me a second, Adam. Your genomic sequence, that is, you see this box here that can you see what my cursor's pointing to, Dipper? Yeah. Click on that. That's what you need to select wherever you put your genomic sequence, the reference sequence, either from what I put on Dropbox or Blackboard or NCBI, it doesn't matter, wherever, right? Valeria, there may be another way that you open stuff on Macs. I don't know if there's something different that you need to do. I'm sorry I can't be more help. I've almost never used a Mac. Now, Dipper, if you now add your sequence of interest, right, from those good yellow sequences files, right? So just click any one of these that hasn't had anything done to it. I don't know. Maybe I'm being a little bit wild here, but let's try this one. So you should have two different sequences here. One is your reference sequence, yellow wild type or yellow.fa or .gb, whatever, it doesn't matter. And then a actual sequence file that you got from those good yellow sequences, right? Have you done that? Then click map, and that should, oh, you got to be kidding me. All right. Curse you, Eugene. All right, so doesn't like that one. Where's one of the ones from GenBank? Pretty sure that's the GenBank one. Yeah, that worked. So try using the one that you downloaded from GenBank. Right, so that's from the, let's go back a few steps. Okay, when you have the yellow gene for Drosophila open, right, you click on this. Pepper, shush. And then the send to file, create file, or actually you can use the one from the Dropbox link that I sent, if you can save that to your computer. That one seems to work. I don't know why the one that I have doesn't. Who knows? Actually, no, that's a wild type new one. So just use the one from MCBI. Give that a shot. Because that seems to work quite nicely for some reason. As soon as you come back, she doesn't want to know me. Sad. Yeah, it's all about the mama. I guess so. All right, give me a sec. Dipper, I'll take you back here. So did you find this page? The Y yellow Drosophila melanogaster page. Okay, so scroll down past this stuff, which we already used last lab. Keep going. And you should get to something which says NCBI reference sequences. It may be that it doesn't like FASTA files for sequence alignments. Yeah, go to GenBank, go up to send to, file, and then create file, and then save that to your computer. And you can give it whatever name you want, just call it sequence, it's fine. Save that and use that, okay? Now if we go back and try another one. No, you don't want to open it, just leave it on your hard drive. But as soon as it's there in that folder that you've got all the other stuff saved to, go back to Eugene, click on Sanger data analysis, and then map reads to reference. So when you get that box open, hit that one. I think you're doing all of this already. I think the issue is the sequence that I saved as a FASTA sequence. Oh, you must have just got the rain that we had, I guess, Robert, because it just chucked it down here a few minutes ago. Yeah, I think it doesn't like. Now, I'm making myself notes here because otherwise I'll forget by next semester. Oh, that's wrong. Get rid of that. I just want to see if this one works. It should do. Yay, happy dance, happy dance. That should work for you, Dipper. If you still are able to have internet, which hopefully you will. How's everybody else doing? Sorry for this is basically the peculiarities of Eugene. It's kind of what happens when you don't pay for software, basically. I think try it with that sequence you just downloaded for the reference and one of the unmolested good yellow sequences, and it should work. I just did it on mine and it was a happy camper. So give it a shot. Oh, by the way, Adam and Dipper, you can send a chat to other people specifically, I think. Actually, I'm not 100% sure. Should be able to, so you don't have to share that. Oh, you can't? Oh, I'm sorry. I don't actually know how to enable that. Well, you could send it to me and then I can send it to him if you want. Did you have success, Dipper? Please say yes. I'll be ever so sad otherwise. What do you mean, kinda? You either did or didn't. What's that? Print screen? Oh, that's pretty funky. Oh, okay. So you've actually opened the sequence. That's cool. That's totally fine. Now that means that the sequence you downloaded is good, right? That's awesome. Now we need to go back. That's actually pretty cool that you can do that. You're way more technologically savvy than I am. Now go back to Eugene, right? You don't have to open the sequence file that you just opened. You have to go through these steps to map the sequence, right? Go to the top, hit tools, right? I'm going to do it slowly so you can do it as I'm doing it, okay? Go down to data sequence or data, Sanger data analysis. And then select map reads to reference, right? How's that all going? Oh, doesn't get angry. Come on. So have you opened this box? Do you have that open in front of you? Yep. Okay, so wherever you saved that sequence file, go there and click OK, right? You should have something that looks like, it's obviously not going to be Matt's work teaching blah, 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 genetics lab, but wherever you saved that file, that's what it should show up here. Do you have that showing? Or is it making angry faces at you? Good. I am a master of internet support. Okay, so go down here, click add. Right, that should pop up another box. Now, wherever you happen to have good yellow sequences saved, obviously it's not going to look like this. Click on that and then select one of these that just has this name, right? Not sequence after it. Those would be the edited ones. You don't want to use that. So let's say this AFCG WENS 57F. Yay! That is super, super awesome. I'm very happy. Cool. All right. Mother Nature and State of Texas Power Board cannot hold us back. Oh yeah, color code letters. Right, so what you're looking at the top, and hopefully everybody else has got the same too. If you haven't, let me know and I'll I'll help you out, promise. So anyway, this is your reference sequence. Now, if you look down, hopefully you should see this box, right, above all that stuff down there, which tells you what you did wrong. And essentially that arrow is telling you where your sequence maps to. So we're going to scroll a little bit to the right, like so, and now you see your sequence pops up. You got that, Dipper? Should be around base 100 or so. Oh yeah, that's cool. No, go away. Not you lot. Computer telling me to do something. So this is our it's basically got rid of all the N's. Those probably shouldn't be there, but they worked anyway. And that will show you, it should be kind of base by base match. Aha. Apart from this one here. So let's keep going. Right, all the way to here. And this is some kind, oh, this is some nasty ass looking sequence. So that's why it didn't match very well. So ignore that. Now, if you do that for a few, not just one sequence, let's see what it says in the handout, just so I'm not jumping ahead and confusing you all. All right, questions 10 to 13. So this is kind of neat because what we have here, okay, stretch, prepare myself for a teaching moment. Crack knuckles. Oh, Adam, don't cry. Make yourself a stiff drink of coffee or something. So essentially what this is doing is it's saying that we have a difference from this sequence up here to the sequence of what we actually had sequenced, right? So I'm going to answer your question. You're in a second, Dipper, because anyway. Now go back to the previous questions in that assignment. See if I can get back to there. Can't remember which number it was. Okay, that'll do. Question 8. Okay, Dipper, I'll just remove that from the chat file before I post it online. Your email address, that is. Where does exon 1 start? So the 5' UTR is from base 1 to 171. What base number is the start of exon 1? This is what I want to do right now. Lie on the couch or a hammock and have a nap. That'd be very nice. The struggle. But it's really cool though. Thank you all of you for being patient while I figure this out. Because now I've got extra things written on a post office receipt that I need to add to that file to make sure that next time I teach this, I don't go, Eugene. And go, I knew there's something I need to do different, but I can't remember what it is. Anyway, back to the question. What bases correspond out of these to exon 1? Actually, we could even look up. Oh, actually, that's not super helpful. I'm not going to look at that. Let's look at this instead. Yeah, it's 172 because that is the first base of exon 1. Start codon is base 172 to 174. So from the information that we got off of Flybase, what's the mutation? What's the change for the Y1 allele that results in yellow flies? Yeah, it's an A to C base change. So you are 100% correct, Dipper. I didn't want to just like give you praise and congratulate you too prematurely. But yeah, you are 100% correct. Now, the reason why I didn't want to give you, too much of a pat on the back is that this is just random chance based on which sequence I picked. So some of these are wild type from wild type flies. I think you deserve a bigger pat on your back than that. Some of them are actually from yellow flies. So if this was a yellow fly sequence, you would not see this A here. It would also be a C. So this here is your start codon. So the Y1 allele is an A to C change in the start codon. In other words, ATG to CTG. Oh, orange at the end, kind of over here. That means those dashes are spaces that the program adds to fit the rest of the alignment to it. I think that's actually a question in Blackboard. So essentially, it doesn't mean that the reference sequence is wrong. It means that these two A's, if you actually look at the electropherogram or the chromatogram, whatever you call it, I wouldn't even say there are bases here, right? This looks like gibberish right here, but the program doesn't know that. It doesn't do like a quality check on based on the chromatogram, and so to make it all fit together, because obviously these ones fit, it adds a couple of spaces to one or other of the sequences to make them line up properly. So these aren't like mutations or other things. It's typically from a sequencing artifact that leads to a slight mismatch between the two sequences. Cool. All right, what were the other questions? So we know what base we should be looking at, right? Nudge, nudge, wink, wink. What's the next question? We also should be able to answer that, right? If the start codon should be an ATG, the CTG sequence would have functional protein or non-functional protein, or any protein at all, actually. Should help you answer that question. Okay, so based on what you know about the start codon and its importance in translation, you should be able to answer this question. Okay, so now what you need to do I think you do, Dipper. Sometimes it just takes slowing down a little bit and just thinking through stuff. All the information you need is already here. You just need to kind of think through it. Okay, so now what we need to do is this part of that mapping? Let me just double check. Yeah, okay, so now we need to do a little bit of multitasking. All right, so we know what bases this aligns to, right? So I'm just gonna write this down because I can't remember off the top of my head. So let's say the last good base is 446. Right, and let's scroll over here. Shunk. First one is 102. Okay, tried what already, Adam? Being bold? Adam is silent. What happened, Adam? Anyway, so what we can do is either use the, that's right. Hang on, let's open up. Bum, bum, bum. There we go, that looks nicer. So, that's actually not super helpful. Let's just go back to question 8. And I think we'll call it a day after this because I'm getting pretty wiped, and I'm sure you all are. We can do the multiple sequence alignment on Tuesday. I think otherwise you'd be not super happy. All right, so kind of going back to this. Sliced fried bacon. Bacon, that'd be nice right now. Get some out of the freezer. Sorry, distracted. Mine just feels like it's dribbling down my ears. That's why I keep wearing the headphones. It's just to keep it in. Okay, so look at these numbers, right? 5' UTR is 1 to 171. We already know that exon 1 is, where are we at? 172 to 409, right? So let's just write those down. And I'm not particularly bothered about telling you this stuff because most of you have probably already done this already. And it's not hard to figure out, right? If you just kind of think through what's going on. Nope, that's 5' UTR. What's the very first thing in a eukaryotic gene? It's the 5' UTR, right? Yeah, so that's got to be the first thing. You know, you can pretty much work it out just from nothing else, really. Oh, actually, yeah, that would work. I was thinking something else. Pardon me. So you can see it here, right? So here's our mRNA, the bit in the weird kind of greenish color. And the bit that corresponds to protein, our coding sequence, is the funny kind of turquoisey cyan fuchsia sort of color. Maybe not fuchsia. I'm not very good with colors. Like that weird kind of bluish color. So that's telling you the 5' UTR has to be the first thing, right? Because it's not translated, i.e. UTR. And then, therefore, first exon, exon 1, has to be after that, which is 172, which we already know because the start codon's mutated, and that's base 172 to 409. Obviously, there has to be an intron between the exons. You can't have an intron after an exon, and then nothing after that. So that's what, 410 to 3108 is absolutely huge. Now look at your map. Let's go back here. No, not that bit. Here we go. Here we go. So go with chat box in one eyeball and your map on the other eyeball. Robert, you've got to use one eyeball for each. You can't change your focus. It's just not going to work, right? It's a skill, but yeah, you'll get there. Anyway, You can use both eyeballs at the same time if you want. So our sequence read starts at 102. Where does our sequence read start out of those three things? What feature of the gene does our sequence start in? Does it start in exon 1? Does it start in the intron? Is it exon 2? Is the number 102 higher or lower than the number 171? Yeah, good stuff, Desiree. It starts in the 5' UTR. So it tells you it has a chunk, 70 bases, give or take, of the 5' UTR. It's very cute watching my puppy go downstairs. Hello, Trouble. Now it ends. Hello, love. Oh, she's in bitey mode. I'm gonna take my hand away. Are you gonna start? Yeah, she's gonna start. She's trying to get our other dog to play with her. Anyway, let's finish this quickly before Trouble interrupts proceedings. Oh yeah, that'd be nice. I'd love to eat a pint of ice cream. So just to finish up, right? Nearly there. Trying to focus. Sequence read. No focus, only ice cream. I'm trying. Like, gotta keep, just can't look at puppies. All right, this ends at 446. Now is 446 higher or lower than 410? She told you so, didn't she? She's being a furry pest, is what she's being. She can come downstairs on her own, love. Okay. 446. The end of the sequence that we're using, we're mapping. Not the reference sequence, the sequence we're mapping. Is that number higher or lower than 410? Exactly. So the PCR product has to include a portion of the intron. Now, if we have a bit of 5' UTR and a bit of the intron, what's the bit in between those two? You were quicker than I was. Exon 1. Right, so that's the answer to that question. Now you can see why it's useful to map the sequence that you've got to your reference, because it'll tell you, like, what am I actually looking at? Which bit of the gene am I working with? All right, I think that's more than enough for today. That was pretty tiring for everybody, I'm sure. So good job, everybody. Thumbs up, pat on the back for everybody that contributed and just was here is awesome too. And we'll finish mapping not mapping, we'll do the sequence alignment Tuesday. It shouldn't, touch wood, cross fingers, take too long, but we have plenty of time to do it if we need it. Sounds good? All right, I'm going to lie in a darkened room for a little while and then have a cup of coffee. You can all go and eat ice cream, go for a walk, whatever, right? Clear your brain out, kind of stare at a wall for a little while, probably wouldn't hurt either. Reevaluate life. Anyway, have a great weekend, everybody. I'll see you on Tuesday. We'll finish up essentially the course, right? That's pretty much it. So yeah, have a good weekend. Study hard. I know it's not easy. You know, there's a lot of things to cover. If you got any questions, let me know. I'm going to be away for Friday and Saturday, so I might not get back to you if you send me something Friday afternoon or during the day on Saturday, but I'll be back here on Sunday, so I'll be able to help you out with whatever needs helping out with. Cool. All right. Take care, everybody. Thanks for your time. It's been good to figure stuff out. Catch you later. You? Yeah.